All right, guys. Today we are going to be talking a little bit about cancer and the cell cycle. I'm sure that last time you watched the notes and you left wondering what happens when our cell cycle regulators are not working the way they are supposed to. And the answer to that is oftentimes that the cells can form cancerous cells. So, when we talk about what a cancerous cell actually is, it is a cell that does not respond correctly to the external or internal regulatory signals that would usually control the timing of our cell cycle. So, when we think about our normal cells, how they actually respond, they're going to stop dividing in response to a lot of our external cell signals or external environmental cues. One of those environmental cues is going to be the absence of growth factors. If you guys remember, we had talked about the epidermal growth factor, right? As a ligand that actually bonds to a cell surface receptor and starts a chain of events that allow the cell to divide. Okay. Another thing that would actually keep our normal cells from dividing would be cell crowding or contact with neighboring cells. So when cells notice that they're too close to one another, they will usually send signals to stop dividing. That is a process or a term that we call density dependent inhibition, right? The cancer cell, or sorry, the normal cells are going to have their division inhibited based on the density of the surrounding cell. Another thing that a normal cell would stop dividing in response to would be the absence of a connection to some kind of surface in our body. That surface is the extracellular matrix. Actually, needing a surface to bind to is something that we call anchorage dependence. And when we think about the three things that we just listed, those cues that normal cells respond to, our cancer cells are actually going to ignore those signals and continue to divide, despite the fact that the external conditions may be telling them not to. One thing to keep in mind about our cancer cells is that it's not just going to be one piece of our regulatory mechanism. It's going to be multiple regulatory mechanisms that build up over a period of time that will actually lead to cancer. So, looking at our image right here, we have this kind of hypothetical series of mutations that might lead to cancer. We might have an initial mutation that's going to inactivate one of our negative cell cycle regulators. So something that usually stops the cell cycle from happening when it's not supposed to is mutated, and our cells continue to divide. Because of that initial mutation, maybe we have another mutation that occurs later on, and it leads to an overactive positive cell cycle regulator. So something that tells the cell it needs to divide even when it's not supposed to. Over time, that accumulation of mutations is going to be what actually leads to your cancer cell. So it's not one mutation, it's not two mutations, it's not just any mutation. It's a series of the right mutations that will actually lead to cancer. And that's why we think of it as something that occurs over a long period of time or a disease of old age, because a lot of times it takes time, right? We have years and years where those mutations are accumulating before we end up with the formation of a tumor or the development of cancer. Okay. So, just like we kind of mentioned, positive cell cycle regulators and negative cell cycle regulators, those are going to be the things that are mutated in our cancer cells. So, we have what we call oncogenes. Those are going to be our positive cell cycle regulators. And we have tumor suppressor genes. Those are going to be our negative cell cycle regulators. And those are going to be the proteins that are not functioning in our cancer cells. So we're going to start with our proto-oncogenes slash oncogenes, all right? So our proto-oncogenes slash oncogenes, like we just said, are positive cell cycle regulators. that are going to be actually overactive in our cancer cells. Okay. Proto-oncogenes are going to be the normal form of our genes. So when these genes are working optimally or the way they're supposed to, we call them proto-oncogenes. And then the oncogenes are going to be the gene in its mutated form. So there are many examples of positive cell cycle regulators that could be impacted in cancer. But the example that we're going to talk about today is a protein and a gene that we call RAS. Okay, so if we look at our RAS protein, I'm just going to write this on the side, is actually a G protein. If we remember how G proteins actually work, they are active or on when they are bound to GTP, okay? And they are off when they are bound to GDP, right? And usually there's some signal that's turning that G protein on or off. Okay, here in this example right here, we see our normal RAS is going to usually respond to a message sent from this membrane receptor or growth factor receptor, okay, which tells the G protein to swap out the GDP for GTP and turn that RAS on. That's going to lead to our cell dividing and proliferating in response to the growth factor. Okay. Now, if we have no growth factor, we're going to see that our RAS remains off. It keeps that GDP. It's not receiving the signal that tells it to trade out that GDP for GTP. And we have no cell division or no cell proliferation. Now, when we have a oncogenic form of RAS, so this is the proto-oncogene here, the normal form, the oncogenic form is going to trade out that GTP for the GDP, and it's always going to be on regardless of if there is a signal present or not. Okay, so that mutant RAS is always going to be bound to that GTP, and the cell is always going to be receiving the signal, okay, it's time to divide. Okay, so if we think about in your HHMI activity, you saw this brake pedal, gas pedal kind of analogy. You can think about the oncogenes as being like putting your foot or a brick on the accelerator and just telling your car to go, 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 even if there's a stop sign, even if there is a traffic light, okay? There is nothing you can do to stop your car from going in that scenario, just like there is nothing you can do to stop cell division and proliferation in this scenario. Now, one thing to keep in mind here is this is not the only example. This is not an example you have to have memorized, but just so you can see how the mutated positive cell regulators will affect the cell, okay? Another way this could work is you could have maybe a receptor that's sending signals all the time to the cell to divide, even though it's not bound to the ligand. Or it could be a cyclin that's just produced at really, really high levels, even though it doesn't need to be produced. Okay, so another example could be either of those things, not just this RAS protein here. Okay. The next thing that we're going to talk about then is our negative cell cycle regulators that can be affected in cancer. And those are what we call our tumor suppressor genes. Okay, so again, just like I said, tumor suppressor genes are negative cell cycle regulators that are either less functional, so they can still be there, but sometimes they're just not functioning the way they're supposed to be. So less functional, or even absent or inactive in our cancer cells. Okay. So these are really, really important, and they actually think that tumor suppressor genes are involved with the development of almost every single type of cancer, specifically one that we're going to talk about in a second called P53. All right. Now, when these are working correctly, these tumor suppressor genes are going to prevent tumor formation by stopping the cell cycle. So a normally functioning tumor suppressor gene will act kind of like the brakes on your car. Okay. And it will tell the cell, hey, stop dividing, something's wrong. Our big example here, all right, is P53. A lot of people call this the guardian of the genome, because it's going to be involved with making sure that damaged DNA doesn't get passed on to our new cells. Okay, so here we can see our normal P53 is going to be activated at the G1 checkpoint, right? Because remember, the G1 checkpoint is where we check for DNA damage before we go and replicate the DNA in the S phase. Okay. If we see that DNA damage is present at that G1 checkpoint, P53 is going to be activated. It's going to stop the cell cycle. The way it does that is by making a CDK inhibitor, which we can see here. Remember, we need those CDKs to kind of activate our phosphorylation cascade and control the cell activities that will get us to that next phase. Okay, so it's going to make a CDK inhibitor that's going to inhibit that cyclin-dependent kinase. It's going to activate repair enzymes to probably try to fix that DNA damage if possible. And if the DNA damage can't be fixed, it's going to tell the cell, like, hey, time to go. And it's going to trigger apoptosis. So that's only if repair is impossible. We're going to have that programmed cell death. So that we're not passing on mutated genes to the next cell. Okay. Now, if our P53 is not functional, all right, we're going to still recognize the DNA damage in that G1 phase or at that G1 checkpoint, but we're no longer going to activate that P53. We can't bind to the DNA, and we can't perform all of those activities that would occur in our normally functioning cell. So we would not have P53 producing that CDK inhibitor. We would not be able to repair the damage in the DNA, and we actually wouldn't see that apoptosis is being triggered. So what happens is, even though we don't want to pass on this DNA, this is not what we should be doing, we make cells and we pass on the DNA. Okay. Now, once we pass on that mutated DNA, those cells are also going to divide. We're going to inherit any of the genetic mutations that were in that cell. So we start to see this kind of cascade of more and more mutations occurring over time. Okay. If we go back to that idea again of the normal P53 being like the brake, our non-functional P53 is like we've cut the brake lines. Okay, so the thing that controls the brakes, we've cut that off. So even though we're in a car, we're trying to stop going, we can't stop. Okay, so in contrast to that go signal, like putting a brick on the accelerator, like we saw with our positive regulators, we're going to be cutting those brake lines. Okay. If you have any questions, please leave those at the end of this video. They can be questions on the HHMI activity or on the notes themselves. And hopefully this made sense, and I'll see you guys in class.